What is considered a competitive inhibitor? So just to review, these inhibitors are referring to substances or molecules which prevent a reaction to occur between an enzyme and a substrate. And the reaction that would be occurring between the enzyme and the substrate would be a catalytic reaction, so catalyst, which helps to speed up a chemical reaction, so speeds up. So the difference between a competitive inhibitor and a non-competitive inhibitor would have to do with the way that the inhibitor is working to prevent this process from happening. So remember, our inhibitor is some sort of molecule that is preventing this process. So we're looking for, is it happening competitively or is it happening non-competitively? So if it's happening competitively, so let's put competitive here, you have your enzyme, let's say there, and let's say that that's the active site where the substrate would go for the reaction to occur. So this is your active site. And let's also make right next to it the non-competitive example. So let's say that this is your non-competitive, and we're going to start off the same way. We're going to draw our enzyme, and we are going to draw our active site. Now, if we look at our competitive to start, we have our substrate that is trying to go in this location where the active site is to form this reaction, this catalytic reaction, right? But if, let's say, in addition to your substrate that could go, we'll just say that it's the shape that fits there, you also have this other molecule that's competing for the same spot. So they both are trying to get in the same spot to have the same reaction, and they're both competing for the same place in the active site. So if you think maybe there's a special seat in the classroom and two students really want to sit there and they're both running together to try and compete to get to the same spot. So you're competing with each other, you're working against each other to get to the same area. The difference between that example of the competitive reaction site, the competitive inhibitor, and the non-competitive example would be that, let's say, when you have your substrate that wants to go into that active site to form the reaction, you have another molecule that goes somewhere else. Goes, let's say, over here, it's able to bind. So it's not binding to the active site, which prevents a full-on catalytic reaction. It inhibits it, not to the same extent as the competitive, but it still does prevent some of the reaction from occurring. So they're not competing for the same site, they're competing for a different site. And that's the difference between your non-competitive example versus your competitive example. So going to the choices here, we see option A says, for a competitive inhibitor, which is the definition, a substance that binds and inhibits an enzyme at its active site. And that's the key word there to know that we are talking about a competitive inhibitor. So this would be your competitive inhibitor. So that would be the answer to this question, because that's what we're looking for, competitive inhibitor. As opposed to option B, which says a substance that binds and inhibits an enzyme at a location different from the active site. So if we look at this example here, the non-competitive inhibitor, it's at a site different from the active site. That would be the definition of a non-competitive inhibitor. So not the answer to this question. Now, taking a look at options C and D, it says a substance that binds to an enzyme at an active site but causes an opposite effect of an agonist. And option D saying a substance that binds to an enzyme at an active site and causes its full activation. So while these are not definitions of competitive inhibitors, these are explaining different processes that we will review as well. So option C is actually what's known as an inverse agonist. So in this example here, a substance is binding at the active site. So similar to competitive inhibition, it is at the active site, but the difference is it is not actually inhibiting the reaction from occurring, the catalyst reaction. It's just causing an opposite or different effect from what we initially would want to see in this enzyme substrate reaction. So this is known as inverse agonist because it causes a different reaction, but it's not actually preventing the whole thing from occurring as we would see in this example of competitive inhibition. So we will eliminate option C. And then option D is saying a substance that binds to an enzyme at an active site and causes its full activation to occur. So in this example, we would not be inhibiting anything, we would be agonizing, so it's an agonist, but instead it's not an inverse, it's actually occurring with its full activation, so this would be a full agonist. So different term here, not what we're looking for this question, so we can eliminate option D. So option A would be correct. The substance that binds and inhibits an enzyme in its active site is competitive inhibition.